It gives me, tremendous pleasure to introduce today's speaker, Harry Greene. Harry requires no introduction, but he's going to get one anyways. So Harry was a former MVZ curator and IB professor. He taught the courses that I teach now, IB 104 and herpetology, much better than I did. One of the many recipients in the MVZ of the Distinguished Teaching Award during his time here, and of course Harry is super well known as being one of the world's, if not the world's, expert on snakes, and so he's published multiple books. He published the book that's become a classic, The Snakes, the Evolution of Mystery and Nature. You might have seen that book. It was extremely well received. One of the awards that it received was the New York Times 100 Most Notable Publications or Books, and it received additional awards as well that I'm not going to remember the title. The Pens.
Whatever.
Yeah, you don't want me to say it. Well, so part of the reason for mentioning Harry's prior book is that he has another book that's about to appear. So the title of that book, which I've just found out about, is Tracks and Shadows, Field Biology as Art. And apparently the MVZ is going to figure somewhat prominently in that book, so that's something we can all look forward to. So today, Harry's, I think, not going to talk so much about his research on snakes, but more about pedagogy and I guess the approach that he's invented in order to teach the Tree of Life to students who otherwise might not be that interested in the Tree of Life. Thanks a lot, Harry, for being here.
Thank you. Thanks everybody for coming. I'm flattered that you would come here on a summer day after the semester's over. So I spent the last 15 years teaching intro bio level courses. I spent 10 years teaching intro bio to non-majors, so every fall I had to walk into an auditorium of 417-year-olds daring me to bore them, basically. About 60% of those students were business majors, and I taught the first semester of a 2-semester course in introductory biology. My semester covered the Tree of Life, evolution, ecology, behavior, and conservation, and the spring semester covered cell and molecular biology and so forth. The last 5 years I've been involved in revamping our core curriculum in the biology major. We went from having intro biosequence to having 5 core courses. One of them used to be called Evolution and was 2 lectures a week, and now it's called Evolution and Biodiversity, and it's 3 lectures a week. And I pushed for that change to the Evolution core course because I think every bio major should know the Tree of Life. I don't think you should be able to get a degree in biology without being conversant in the basic typology of the history of life, the history of descent and modification. So now I teach a 12-lecture segment in which I have 12 lectures to go all the way from the origin of life of bacteria to primates. And I'm not saying that I've solved everything. I'm not saying that what I'm going to talk about is the only way to do it, but I have thought a lot about, made some mistakes, watched other people make mistakes and solve them and so forth in terms of how to teach about the diversity of life. So that's what I'd like to sort of lay before you today. And I just want to hammer this point. I really would like to see someone with a straight face get up and defend having a coverage of biology that does not include the Tree of Life. And yet, as recently as 5 years ago, you could get a degree in biology at Cornell and not know what the word mammal meant, not know that not all plants make flowers, not know that archaeans are more closely related to eukaryotes than they are to bacteria, not be conversant in those things. You might even still think that algae are a monophyletic group, okay? So that's not okay. I don't think you should be able to get a degree in biology, no matter whether you're going to be a biochemist, a physician, ecologist, anything else. You should know the Tree of Life. It's the core of everything. So we hear a lot of talk about how we can leave various things out of the curriculum as long as we teach certain principles. The students can generalize from one thing to another. I don't think that applies to the Tree of Life. I think it's the very basis of biology. It's what evolution explains, and everybody should be exposed to it. So when I go in there the first day, I tell the students that I'm going to make them learn about 145 taxa. So I pruned the Tree of Life down to about 145 taxa. And I've scrambled these 145 taxa on 3 PowerPoint panels. And I show them to them fairly rapidly, so I ask them not to spend time concentrating this. I just want to show them that by the end of my 12 lectures, if they buy into what I'm going to try to do with them, they'll be able to talk about all these organisms. So they'll not only know what all these words mean, here's the second third, and here's the third, at which point they are just already going, oh my God. I'm going to have to learn words like sarcopterygian and so forth. And so I tell them, not only are you going to have to learn these 145 words, but you're going to have to know about 3 things about each of them. And now they're just glazing over. But I have been asking them already, do you recognize any of these, or any of the human pathogens, or any of the model organisms and so forth. And then I tell them, But it's not going to be like this because it's not random. This isn't going to be a memorization exercise. You're going to learn the structure of the history of life, okay? And it's going to be completely logical, completely hierarchical, and because of that, it'll be a system of learning. It won't be a memorization exercise. And that's because we're going to learn it as the tree of life. And I tell them, you know, if you buy into what I'm going to teach you, you can do what I call walking and talking the tree of life. I could put you down here in mammals, for example, and ask you to take me to glaucophytes. And if you've bought into what I've taught you, you're going to take me down node by node through what we've learned, down to the base of the eukaryote tree, up the plant tree, and right here to glaucophytes. Okay? So you're going to be able to go through this virtual space and time thing and walk and talk anywhere in the tree of life. That's what I want them to be able to do. Okay? So I think it's really important to sort of show them where we're going. I emphasize that this is not scala naturae. This is not some linear progression. This is over time diversification. It's Darwin's descent with modification. It's a hierarchical system. The nodes mean something. The terminal taxa are all things we've heard of, like bacteria and plants and fungi and so forth. And these collapse down into here, collapses down into here, or you can take any one branch like the eukaryotes and explode it out into this, and you can take one branch of this and explode out into the plants or the animals, and collapse and explode and so forth. It's all completely hierarchical. It integrates across all biology, molecules to organisms, structurally and functionally across space and time. And I think it really has profound implications, not only in science, but also for medicine, for conservation, and so forth. I don't think there's, I would almost challenge anybody to bring something up within the sphere of intellectual inquiry in which the tree of life wouldn't be interesting. I just this morning started reading a book, an anthropological text, in which the first page says that animals are the only living heterotrophs. Okay? Now that's absurd. That's absurd. And if you knew anything about the tree of life, you'd know that here's an origin of heterotrophy right here in the Opisthokonts, because in fact fungi are the closest living relatives of animals, and fungi are heterotrophic too. So I could spend a lot of time giving examples of this, and I don't have that time, especially since you want me to end on time. I think it's really important, especially in these big lecture courses, and especially when dealing with freshmen, to be alert to the characteristics of your audience, to what they may already know and what they don't know. I think they often have very little prior background, and what they've had was often boring, especially with regard to learning biodiversity. So they've still, sometimes in high school, learn the diversity of life the way I learned it 40 or 45 years ago in college, where you had to memorize whether something was a family or a phylum or a class or an order or whatever. It was a lot of memorization. Many of them are pre-meds, and they're obsessed with grades. They want to know, why do we have to know this? They're very skeptical that this has any relationship to anything that they're interested in. And I think one mistake we often make is to assume that they'll just see that it's interesting, and they'll just think that we're interested. And so I actually think the first lecture is really critical, especially in these lower level courses, in terms of hooking them in. And I believe there's actually been research that shows almost like the first 10 minutes of your first lecture is key to sort of setting the tone for your whole semester. So one thing I do is talk about myself just a little bit, and that's not because I'm an egotistical maniac or anything. I just want to get their attention. I want to convince them that they ought to give me a break. I mean, I'm more than 3 times their age. It's self-evident that I am more than 3 times as old as everybody else in that room. And so they are naturally skeptical of whether I'm worth listening to. Certain things on this, like the 1.89 GPA, tend to draw out their emphasis. And actually a very funny thing happened this last year. Kelly is actually in charge of this course. And so she'd already given several of the lectures. And so when I got down here, I'm married to Professor Zamudio. This guy in the back went, wow! One thing I insisted on is that we have a separate text in this course for biodiversity, so they have to buy an evolution text. I think it's that having to purchase a textbook enhances the impression that this is important. And I want them to have a reference work that they can rely on both during the course and in the future. There's not a single textbook that does exactly what I want for this course and addresses learning the tree of life in the way I want to do it, but I found the thing that comes closest to it are the biodiversity chapters in this Principles of Life book by Hillis et al. And so we went to Sinauer, we asked them, they developed a whole separate, publishers will do this if you have enough students buying it. So we have a separate text made from the 5 biodiversity chapters of Hillis et al. We got to design our own cover for Cornell. By accident, the first year, they put the glossary for the whole intro bio book into my biodiversity text, and this turned out to be an unintended wonderful consequence, because since we've gone to this core course approach, our students don't have an overall biology textbook and they don't have an overall biology glossary. Now they do, and it's in their biodiversity text. And so when I talk to them about the text, I tell them, this is the one book you want to keep for your whole 4 years as a bio major at Cornell. You can sell your biochem text, you can sell your evolution text, but keep your biodiversity text right there in front of you, because in every course you take in biology, you're going to be referring back to this sort of scaffolding of the tree of life. I warn them that the tree of life is in flux, especially at some of the basal levels. We're still learning a lot about these relationships. They're even changing from edition to edition of some of these books. But this is not a criticism of an understanding of the science of life. This is typical. I mean, biochem textbooks get revised too, so just get used to it. Things are going to change. But if you buy in and can walk and talk the tree of life, you will be ready to interpret contradictory things, changes that come up, and so forth. And I remind them that it has a glossary. There are some sort of basic principles that have come out of trial and error of teaching this for 15 years now, and teaching it with other people who had various notions about how to do it. So one of the things we've learned is that, at least at the level of 12 lectures and working with freshmen, too many taxa is a huge mistake. And it's really hard to do that. Imagine having to prune the whole tree of life down to 145 taxa. Imagine having to leave snakes out. I mean, it's hard, okay? It's really hard, but you've got to do it. If you put 200 taxa up there, their eyes glass over, they get bored, they get discouraged. I think it's sort of counterproductive. So it's very important to be very strategic in terms of local goals, local pedagogic goals, particular things you want to emphasize. I think if you've got pre-meds, you really want to have some pathogens in there. And with the pre-meds, I tell them, what are you going to do? You're a 21st century doctor. You diagnose your patient with Giardia. And they go, oh, really? What's Giardia? And you say, oh, it's a bug. Or it's a germ. I mean, come on. Really? That's good enough for a 21st century physician to tell somebody Giardia is a germ? Or worse yet, you're going to tell them it's a bacterium, which is not, or a virus, which is not. Okay, it's not an animal either. So you better know what Giardia is if you're a physician. And this is the framework with which you can talk about it. I think whenever possible, monophyly is extremely important. I'll have a couple examples of that. I think that we're way past time to dispense with ranked classifications, especially at higher levels. I think it's a waste of students' time to learn whether something is a class or an order or a phylum. So I teach biodiversity completely phylogenetically as an intended classification. I don't make people learn the names of any ranks. I make them learn the names of what they mean. So I don't really care whether they know whether mammals are a phylum or an order or a kingdom or a class or whatever. I want them to know what a mammal is, who are the mammals, who are the closest relatives of mammals, and what's the next largest group that includes mammals. So I'd rather they devote brain space and time to that. And of course, in prioritizing these taxa and setting up the coverage, we like to emphasize nodes that have really interesting characters, emphasize descendant modification, and I'll show you one or two examples of that. So here's the way I learned it in the early 60s as an intro bio student. We didn't have domains back then, but we had all these others. And we were told that this was the Linnaean classification and that it was evolutionary and that it was hierarchical. Now we know that that's only vaguely true. I mean, it's true to a certain extent, but it doesn't go nearly far enough. And so in fact, it doesn't reflect evolutionary history very well. And if you're skeptical of what I'm saying, here's just a tiny subset of the Linnaean hierarchy of life. And here are these classes within the subphylum Vertebrata. They're all equally ranked. Okay, everybody knows what these are, right? The jawless vertebrates, the cartilaginous fishes, the bony fishes, the amphibioreptilobies, mammals, right? But look at all that's wrong with that. First of all, it misses out on some major events in the history of life. No group there for the origin of jaws. No group there for the origin of four limbs. No group there for the origin of amniotic eggs. That's all left out of what I learned as a freshman intro bio student in the mid-60s. All kinds of paraphyly. Some of these fishes are more closely related to these animals than they are to other fishes. Some of these reptiles are more closely related to these than they are to other reptiles, and so forth. elevation of some groups because they have wings but not others. I mean, if wings are so important, why don't we have a class Chiroptera? Got a lot of bats. Might as well elevate them. They fly. So I think there's got to be a better way, and I think there really is a better way, and that's to use an indented classification that exactly reflects the history of life. So here's an indented classification of the really base of the tree of life, in which, interestingly, there's no name for eukaryotes plus archaeans. And that's probably for at least one reason, and that is you had all this lateral gene transfer. So a lot of crazy stuff was going on down here early in the history of life, and so it gets a little problematic just who's got what down here. But there is not a name for archaean plus eukaryotes. So there's two ways to go with that. You can either think of an unnamed taxon that includes those two groups, or you can still use the word prokaryotes to talk about archaeans and bacteria. And that, of course, would be a paraphyletic group. Here are most of the major lineages of eukaryotes. There's still a lot of controversy about some of this. It's an ongoing resolution, but I think it's pretty exciting that we really are finally getting to know these lineages, and we can get away from just calling them all protests. Even at this level, start to run into prioritization problems. So there's some people I'm really glad are not in this room because I've left out the rhizarians. Those are the forams. Okay, so if you have somebody in your apartment that works on forams, you're probably going to want to leave the rhizarians in. I got away with leaving them out. But here are the main lineages of eukaryotes. It's not too many words. And I tell the students at this point, if this was a really easy class, I could just make you learn 13 taxa and we'd be done. All you have to know is that every single living creature falls somewhere in here. Mammal, in here. Snake, in here. Mushroom, in here. You know, fir tree, in here. I mean, you can do that, except we're not going to let you get away with it. We're going to make you learn about 145 instead of 13. I talked to them a little bit about Paraflex Taxa because there are so many kind of hanging around out there. They're gradually getting dropped out. So I think it won't be long before we won't have courses called invertebrate zoology anymore. But here are some Paraflex Taxa. Eukaryotes, which are basically bacteria and archaea. Protists, which are basically everything that's not a multicellular plant, animal, or fungus. But it always includes actually some of the basal lineages of these other groups. Microbial eukaryotes, basically eukaryotes that aren't multicellular and tiny. Protozoa. I mean, I took a whole course in protozoology as an undergrad. I took a course in algology as an undergrad. You know that algae, as I learned it in intro bio, included cyanobacteria, brown algae, red algae, green algae, all members of different lineages. It's really amazing how much we've learned since then. Invertebrates and bony fishes. I predict that we're going to come to call osteichthyes bony vertebrates before long and get over that. I'm just going to show you a few examples of the way we cover some of this. So here's the basal tree where we're emphasizing bacteria. In this set of 12 lectures, for two reasons I don't spend a lot of time on non-eukaryotes. One reason is it's obviously not my competency, so I'd very quickly get bogged down in my own ignorance if I had to go in this very much. And the other thing is that the students are not as familiar with diversity in these smaller groups. They don't sort of have mental images of them, and they're going to cover them in other courses like biochemistry and genetics quite a lot. So we go into some of it. These are the groups of bacteria I make them learn, the spirochetes, chlamydia, cyanobacteria, and proteobacteria. For various reasons, I picked out these groups. I put little symbols by pathogenic lineages that have human pathogens in them. Interestingly, there are no pathogenic archaeans, and that's a subject of theoretical discussion, why there are no pathogenic archaeans. Just to give you an example of how we might cover one group, here's the eukaryote tree. So once again, here's our fungi and our animals, our sister taxa. And we don't cover rhizarians, but then we just sort of go through and lecture, assign readings and so forth, and cover these groups, the alveolates. And I make them learn just a few kinds of alveolates, the dinoflagellates. We talk about dinoflagellates some. The ciliates, talk about them a little more, a little bit of ciliate diversity. Use a really famous ciliate, the paramecium, to talk about some of the structural characteristics of this organism. So, to my mind, another value of having it be a required part of the curriculum is that depending on what people emphasize in their undergraduate careers, this may be the only organismal biology they get. Okay? And so this may be the only 12 lectures in which you really emphasize the organisms itself and lineages of organisms with lineage characteristics. So I stress that in going through this. So here's an example where you can talk a little bit more about the detailed biology and structure of a particular organism. We just can't get away from the pre-med, so we try to, you could call it pandering, but I think it's just playing to what fascinates them. So I often have a clicker question that asks what the deadliest animal in the world is. And I have things like hippos and crocodiles and so forth on there, but actually malaria kills far more than any of those vertebrates. Just to show you a little bit more about the breakdown of the prioritization, and I guess I can't say enough, you can argue with all these priority choices I made, so you might choose something else. But when we cover plants, and planty, by the way, has been redefined in various ways, so I may have Brent throwing something at me or not in a minute, but in our scheme, we use planty to include all the way back to glaucophytes. So we make them learn planty, we make them learn glaucophytes, red algae, green plants. Within that, we defer to a paraphyletic assemblage of several basic kinds of so-called green algae, and then we have land plants up here. I just want to say one little thing. There's so much I don't know about all these organisms, and it's kind of cool to have to teach things that are new to me. Why would you pick glaucophytes to include in this? It's only a few species. Well, it turns out glaucophytes have a molecule called peptidoglycan. You know where they get their peptidoglycan? It's in their chloroplasts. You know who else has peptidoglycan in their chloroplasts? Cyanobacteria. So these are the only eukaryotes that retain in their chloroplasts the signature of the primary endosymbiotic event. Okay? So there are little things like that you can pick out that are really cool, that illustrate descent with modification. And so some of the times we prioritize a taxon because it's particularly interesting from a point of view like that. We go down, so now here we are in the land plants, and within them, again, look at, it's so hard to leave out things. How can you leave out equisetum? Such a cool plant. How can you leave out equisetum? But you just have to cut stuff and cut stuff and cut stuff to keep it down to about 145 taxa. So you might recognize, as we used to talk about something called bryophytes, and it turns out that's not a monophyletic group. So we talked about liverworts, mosses, vascular plants, ferns, seed plants, and then of course within them we talked about gymnosperms and angiosperms and so forth. For every lecture, I have a slide like this right at the start because they're so obsessed with what are you really wanting us to know. I mean, they just want, they want you to tell them, what do I have to know? And I've decided to go ahead and tell them what they have to know, and then hope that that sort of relaxes them enough that they'll soak up some other stuff that I talk about that's not required. So I always have a slide that this is at the start. So this is the one for animals that tells the required taxon names for today's lecture and tells the required terminology, the required things they have to learn about those taxa for today's lecture, and has the reading assignment for today's lecture. I have a slide like that at the start of every lecture. We're going to have fun. You're going to get to graduation. I'm rushing through this. So you're going to make it. Another example of going down through this, here's the amniote tree of life. And there's a really interesting macroevolutionary pattern associated with this. We know a lot about the reproductive biology of all these organisms, including the reproductive biology of extinct archosaurs. So there are a wonderful array of fossilized nests, females with eggs, and so forth. We know a lot about the reproductive biology of extinct archosaurs. And it turns out in this whole array of amniotes, there are no viviparous turtles living or extinct, no viviparous rhynchocephalians living or extinct, no viviparous crocodilians living or extinct, and no extinct archosaurs, including 10,000 species of living birds, or extant archosaurs that have evolved live birth. Live birth is apparently evolved only once in this lineage, at the node for marsupials and placentas. Live birth has evolved over 100 times just within living squamates. Okay. So there's an incredible disparity, an incredibly interesting macroevolutionary pattern here in terms of starting with the invention, so to speak, of the amniote egg, the diversification of life, lineages in which no matter how much diversification they do, they never make viviparity. They make it once in our lineage and then 100 times here. What an incredible thing. You know? What an incredibly interesting pattern that cries out for further study, exploration, and so forth. Then we can talk about it a little bit in detail. It turns out a lot of biologists don't know there are still Agripperous mammals. If they do, they think of it as laying a reptile egg, which is kind of a funny concept, have a mammal lay a reptile egg. It's really appropriate to call it the amniote egg. And of course you can talk about the natural history of these wonderful organisms. I mean, what's cuter than a baby platypus? That always gets you. a little bit. I talk to them a little bit about human reproduction, and I remind them that the key elements of placentation are based on characteristics of the amniotic egg, the amniotic nutrition, vascularization, and so forth. So here's a cutaway of an amniotic egg that they've already seen earlier when we talked about the origin of amniotes. And then here's a figure from the same textbook about the human placenta, this little stylized human fetus, placental connections here. And I tell them that, although this appears to be a fairly complicated thing, and in fact at some level of discussion it would be fairly complicated, at a simple level it's simply the interdigitation of the circulatory systems of the fetus and the mother in such an intimate, complex way that you can exchange nutrients, waste, and gases between the two. And if you look at the structure of this wonderful set of tissues right here, you'll see these words like amnion and chorion that you also saw way back here when we talked about the origin of the amniotic egg. So it's a nice example of assembly modification. Does everybody know what clickers are? Or should I explain what clickers are?
I think you should explain.
I should explain? Okay. So I'm a technophobe, okay? I have an old iPhone that Kelly gave me, so I am technophobic. But I love clickers. I think they're just wonderful, at least for these really big courses. The way they work is that every freshman gets issued a thing that just looks like a little remote for your TV. It has A, B, C, D, E on it, and an on button. They register that with the CIT office. The classrooms all have a receiver. There's a program that goes into your laptop that manages the whole thing. And that if we were in Kennedy Hall where I lecture and I put this up, I would hit a button and you would see a little clock start spinning up here. And I would have set it to stop at a minute or 2 minutes. You would see a little counter that starts going 1, 2, 3, 4, 47, 329, etc. as people pointed their clicker towards the receiver and gave their answer to the clicker question. And then when the 2 minutes were up and I said stop, I would hit another button and a bar graph would go up that shows the frequency distribution of the answers to this clicker question. The most obvious advantage to that is you right away see if a lot of people made the same mistake. So if you give a really easy question, you'll get like this one big bar, 98% got the right answer. If you give a hard question, you'll often get like 50% got it right and 40% got the same wrong answer. And that right away tells you something that you need to straighten out with the class. And I got on that right away. The cooler thing is that making an unambiguous multiple choice question is challenging for some of us. And if you have bright students, they will challenge you. And so I'd go up there and here'd be 50% and I'd look and I'd say, well, that's pretty funny that 40% of you got this wrong. And a hand goes up. And this woman says, I don't think that's wrong. And I go, really? Well, stand up and tell the class why. And so they'll stand up and she goes, well, on page 222 of our textbook, it says it's sexual selection, blah, blah, blah. And I, okay. And I turn to my TA in charge of the clicker points and I say, Anna, we're going to count B right also, and the class applauds. And that turns out to be an active learning moment. Now the best thing was once I actually inadvertently wrote a multiple choice question in which all 5 alternatives were plausibly right. Class, one by one, beat me down. And I said, well, this is pretty funny, most of you missed it, this is the right answer. And all these hands went up, and I said, okay, you. And this guy says, well, you're wrong about B. And I go, you're right, Anna, we're going to count B right. Let's go on. And they're, no. And they beat me down, everyone. So you see the way clickers work. And it's amazing. You can have 400 freshmen who are intrinsically shy about talking to a professor their first semester in college, and you can get them arguing with you in a big auditorium. The other thing I figured out was, when we started doing this, I thought, well, I forgot to tell you, they get a point. And they can make a maximum of about 40 points in the semester the way I do it. And so I thought, well, if there's points involved, they can't talk to each other. That would be cheating. I mean, you can't get help if there's points, right? And then I found out it's kind of hard to police this big auditorium of 400 students. But most importantly, one night I was thinking, I could have stayed up all night trying to figure out how to get them to talk to each other about biology. And all I have to do is pay them a point. And so then I made it so that you can talk during the clicker question. So as soon as the clock starts, you see them coalescing into these little clicks. And they form these little clicker question clicks. And so they argue with each other, pointing at the screen, talking, and then they all do it. That's fine with me. I'm happy to pay them a point to talk to each other about which of these is the right answer. By the way, which do you think is the right answer? Click. What's the right answer?
C.
What is the second oldest common ancestor?
Second oldest.
You guys aren't going to graduation until you get this. C is the right answer, right? Anybody want to contest that? Here's another clicker question. Taxonomy encompasses all human pathogens, and the TOL is what? What would be the smallest inclusive taxon that would include all pathogens in the tree of life? B. Life itself. So even though archaeans aren't known to have any pathogens, to have a node that encompasses all the known pathogens, you'd have to go to the basal node, which we call life. Okay. And I end the 12 lectures by talking about where we might go next with all this tree of life we've learned. And I have to tell you, we haven't done assessment in the sort of pedagogical research sense of doing it. But these two courses, first of all, the evaluation scores have increased dramatically since we started doing it this way. So the students certainly like it better than they like the old way. But I have many anecdotes. I mean, I've been walking across campus and had a student come up to me and say, "Oh, Dr. Greene, I had you in intro bio 3 years ago, and I was just looking at a pine tree yesterday and reminding myself that is not an angiosperm." we found a place on campus where you can stand in one spot and see green algae, liverworts, mosses, ferns, gymnosperms, and angiosperms. So you can stand in one spot and see 400 million years of plant evolution. And we tell them where that spot is. And I've had many students come up to me and say, "You know, I thought it was sort of dorky, but I went and did that." And I had to admit that it was cool. So we tell them, you know, you can appreciate nature more by understanding the tree of life. There really is a sort of phylogenetic and sublime aesthetics. All these other courses you're going to take build on the scaffolding of the tree of life, even genetics and biochemistry. It's more obvious with courses like ecology, perhaps. And then, of course, we have all these ology courses that are spread all over campus in various departments, all of which take pieces of the tree of life. And so I'm going to stop with that and tell you that I also teach herpetology this way. And this is actually how I got the idea for this, was teaching herpetology here at Berkeley. In the early 80s, we had 2 PhD students in our department named Jacques Gauthier and Kevin DeCaros. And it was a tough ride for some of us. I mean, I was dragged kicking and screaming. I really didn't get it for a while, and it was painful to get it. But I got it eventually, and I don't like teaching paraphyletic herpetology, you know, so I just monophilized herpetology. I teach it as the biology of tetrapods. I have one whole lecture on mammals. Why would you do that? Because I want people to think about the diversification in the 2 branches of amniotes. So when you contemplate all this diversification in reptiles, why not balance it with what happened in the other branch of amniotes, you know? When you get to archosaurs, why talk about just 23 species of living crocodilians when you can throw in some really interesting similarities with crocodilians by adding those birds for just a little bit of context? So I was already doing it like this, and that's what led me to it. So I'm going to stop there. Thanks for your attention. I'm happy to answer questions or argue about any of this. Geography, both in the evolution module and the ecology module, because the ecology module includes coverage of biomes. So we sort of integrate. Biogeography is, to me, a very integrative thing across topics like ecology and phylogenetics. So we talk about it in both places, but they do get biogeography. Yes, Tracy.
So how do you talk to them about phylogenetic inference? Does that come before, like in the evolution?
That's a good question. So in the intro course, which I don't teach anymore, but in the intro course, I had to give them a very short intro to that before I did this. And actually, I don't think it's, yeah. So one of the things I actually had to get them into was actually thinking about there being a history of life. And by the way, we survey our students with a clicker, and we know that about 12% of our intro students are literal creationists. So I would just start out. I had this as a PowerPoint slide such that you didn't see this part at first, and I talk about, you know, obviously you know about human family trees, and my brother and I have a genealogy. It goes back to Nathaniel Greene, a Revolutionary War general, and you can, you know, I can pay money and I can get that genealogy. And I say, and then I have this friend, Gabriela Parra from Mexico, and she's got a genealogy too. And I say, do you guys all believe that? Oh yeah, we believe that. And I said, well, do you think that Gabriela and I had a common ancestor? And actually, they don't say yes right at the start. And then I go, yeah, you must have had a common ancestor with Gabriella. And I said, yeah, I think so too. I said, where do you think the common ancestor of me and Gabriella lived? Well, they're not so sure about that. And then somebody will say North America, and I go, really? Because this is John Smith. This is the English. This is Cortez and Hernandez and so forth. Oh, it must have been Western Europe that you had a common ancestor. Well, when do you think that was? Well, we don't remember, but it was long ago. So obviously you can pass us back. And I say we do the whole thing for the tree of all life, except the convention is to flip this upside down or turn it on its side. These are terminal taxa instead of individual people. This is the origin of life, etc. And so first thing I do is try to convince them that there's a genealogy of all of life, just like there's a genealogy of us and our families. And then we do a very little intro thing to phylogenetic inference. In the evolution course, there's a whole unit on phylogenetic inference. And they're getting that. They get that before my first block of biodiversity lectures. Bill?
Since you leave out so many taxa necessarily, lineages, did students ever ask you about, well, what about so-and-so? Where does it fit?
Yeah.
Something they know about but it's not in your system.
Exactly. And I should have said this, that I think that's a very important thing to talk to them about because it's inevitable. But the thing is, the farther back you go in the tree, I'd like to back off this. The farther back you go in the tree, the more likely is whatever they bring up is going to be in there. It's just not visibly in there. And now pretty much you could type any, I don't know if it's any, but you could type most taxa into Google, and you would pretty quickly find a higher taxon that it pertains to. So that's what I tell them, that anything you come up with that I haven't talked about goes in that tree somewhere. And so it's a matter of finding its place. Yes, I don't know your name.
Oh, my name is Iman Sylvain. I'm in PMB.
Hi.
So you made it very clear how you get medical students, pre-med students interested. I'm wondering how you get business students really hooked.
Uh-huh. Yeah.
Is there like an entrepreneurial spin?
Yeah. And I you're making me think about which do I think is harder, pre-meds or business students. With business students, I appealed more to their interest in nature and to the diversity of life around them. I do think there are economic implications of biodiversity, of course, and that there will be sort of tree of life connections to that. So that's a good question. It's probably something I could work harder on. Yeah. Thanks. Dale.
That was just what I was going to say. Tell them about endangered species and the costs.
Yeah, yeah, for sure. Yeah, one of the things you could think about, for example, if you get into conservation-related things is, I believe at least, that we will inevitably have triage. We don't like to admit that, but if we don't do it purposely, we'll have it willy-nilly. We don't seem to be willing to allocate enough resources to save everything, and we probably can't save anything no matter how much resource we allocate. So we could just let things sort of slip into oblivion willy-nilly, or we could ask ourselves, are some of these things more unique and more valuable in some sense than others from an evolutionary history point of view? And there's the tree of life. You need to know it right there. Steve.
Do you give them any experience with anything other than virtual organisms?
Yes. So there's not a lab for this course, but there are things called sections, and they meet with a TA in section, and we buy the kinds of things you and I probably saw in labs, like liverworts and planaria, and we get live glaucophytes, for example. You can get them in a little vial. And so we have microscopes. We do show them some stuff, and then we send them out onto campus to look for things and so forth. Yeah. Yes.
So the NSF has done away with the biotic survey program and the PEAT program, and there's no, as far as I know, no natural history division of NSF. So what is your sort of prognosis for the future of funding at agencies like NSF for basic organismal biology and taxonomy?
There you go. There you go. You're so asking the wrong guy, because I've been spectacularly unsuccessful at getting NSF money, and I actually gave up a long time ago. And coincident with giving up, I feel myself just limitlessly lucky in several regards, the big one having been being in MVZ for 20 years. My research is relatively cheap, and my research while I was in MVZ was substantially supported by the Andy Alexander Endowment for field research, as well as out-of-pocket and from other sources. And then 2 people, a Canadian foundation, dropped out of the sky and gave me substantial money for 12 years with no strings attached. And so that's how I did it. And of course, that's not a prescription for other people. and David and Marley and I have been talking about this. I don't know how to do this, but I would like to engender some sort of patronage system in the, like artists in the Renaissance, where people would be patrons of natural history and support individual researchers. And I would claim that the bang per buck is enormous, perhaps compared to bigger grant-supported things. So for somebody like me, for example, 15 grand a year, I can write a lot of nice papers. I would claim on 15 grand a year, and I would claim that's a pretty big bang for buck. And there are a lot of people out there with a lot of money. The people who supported me, every paper I wrote for 15 years acknowledged these people. Every book I wrote, every student I sponsored, and these people sort of vicariously lived through this. So I don't really know how to do this. Maybe you do, but I wish we could get the people who could afford to support natural history in this way. I'm not particularly, from what I can see, NSF is suffering so much in terms of budget cuts and so forth that I don't really foresee descriptive natural history rising to the top in that system. I might be wrong, but I don't foresee it.
When you have an undergraduate, say, who's really passionate about, say, green algae, and it's all about the organisms first and maybe the questions second, do you square with them and say, it's great that I love your passion, but when you get to grad school and beyond, if you want a career in this, funding looks pretty dim?
No, actually I don't. You know what I tell them is I tell them I'd be the last person to say to not work on what you're passionate about. But if you want other people to pay for this and you want people to hire you to teach, you're going to have to be able to put your natural history in a broader context. And that's sort of your responsibility, is to have at least one or two conceptual and sort of technical domains in which you're so proficient that you can make your natural history interesting to other people. I think that's not a cop-out. I think that's realistic. Thanks.
I have a question sort of about biology education. NSF is putting a lot of money into the kinds of stuff that you do, which is the way that you explain biology, I think, is sort of really cool and really new and what we need. And there are biology departments now hiring faculty members whose positions are biology education. And I'm just curious how do you feel about this, and what does the community think about this? The community of scientists, what do we think about this? Is this a good trend, or should we be doing that and losing, different types of you only have so many faculty positions, and so we're getting these biology education positions that are funded. And in my faculty, we have a really good biology education person, but they do really different stuff. They're asking different questions. And it feels like things are changing. I'm just curious what you think about that.
Yeah. So I'm not going to give you a firm answer because I think this is really in flux right now, and it's a very, super interesting question. And maybe a parallel question. You all heard about flipping courses? So this is a topic of much discussion on my campus. And the guy, I think his name is Greene, the Nobel physicist who sort of really pushing this, he came to talk to a lot of us. And so the whole idea of flipping is to get away from traditional lectures and to have much of the actual time of a lecture spent in more interactive things involving the students among themselves, the student with the faculty, actual exercises that they do that start outside of classrooms. So it would put a lot more burden on the student to study, which I think could be a good thing. But it's sort of, and there are studies claiming to show that students actually retain more from that approach than from a traditional lecture approach done by award-winning lecturers. Okay? So my question to him was, this all sounds good, except I think I thought of two ways to do this. You could just survey a bunch of people and ask them what do they remember about their favorite teacher. Or you can even take the dossiers of people who win teaching awards and go through and read all the student commentaries. But I'm willing to bet you that what they mostly don't say is, the person that changed my life was the one that made me remember more on an exam 6 weeks later when I was retested. Okay, which is what is being used as a yardstick for this flipping approach. So I'm not denying that flipping may be a good thing to do, but I'm really interested in what about the inspirational teacher? What about the kinds of things that cause you to get emails 30 years later from people in your classes, Is that going to be lost? And I think this is related to what you asked me, because the people who are actually doing research might be a little more likely to be that inspirational teacher to answer your question if you're the one guy who cares about glaucophytes or something, than a person who's just into education per se. And I don't know that for sure, but I have a concern about that. But this is a very active discussion right now in the university community. You know, I think how we teach classes, especially big classes, is very much in flux. Yes?
Sorry. I was wondering whether when you revamped the way that you were teaching this course and added this phylogenetic framework, if you also changed the way that you were testing or assigning different homework questions and how that was.
Right, that's a good question. And as you can tell from the clicker question, you know, one of the problems if you teach huge courses is you have a literally a grading barrier, so to speak. I mean, you can't grade 400 times 20 essay questions, or at least it's a little bit hard to imagine. So multiple choice is something we use a lot, not entirely, but a lot in these courses. And so then it becomes challenging to design multiple course questions that not only just ask you questions about the tree of life, but actually challenge you. So you're always looking for ways to do things like the second oldest instead of just a simple question, because the way I ask it there required you to know two things instead of just one. You know? So in the art course where they learn phylogenetic methodology, like Tracy was talking about, there are exercises they do online and so forth that have them constructing cladograms, looking at the effect of adding and subtracting characters. So there's a lot they learn outside of class using methods like that. Yes?
I have to ask, if someone on your campus came to you and said you had to turn this into an online course, could you do it? I mean, we're all facing this pressure now.
Yeah. Well, I guess we could.
But I mean, would it work?
That's you know, I'm thinking about developing an online herpetology course. I'm thinking about this a lot, and I'm not sure how I feel about it yet. So if you asked me in 2 years, I might have decided it's hopeless, or I might have decided I figured out how. What do you think?
I think for some courses it's definitely doable, but I struggle with some of the biology courses because I don't see how you can teach a lab or a field section effectively through an online system. And to me, you talk about a student writing to you 30 years later, that's the reason they're going to write to you, not sitting in a lecture theater.
One thing I've thought about with online courses is having real field trips that you would enroll for, but you could only enroll for this field course if you've taken the online course. So you would have all the students, say, going on a herpetology field trip to the southwestern U.S. for 3 weeks. But all the students would already know the framework of herpetology because they successfully completed the online course, Talkin, the tree of life, squamates, for example, or whatever. But I don't have a clear answer for you. I think it's a very interesting question. I'm not sure. Okay.
What about melding the two? In other words, using the online as part of the teaching method, but also have the face-to-face, which seems to me that actually both work.
Yeah. The face-to-face, of course, could only be with a fraction of the number of people that would be worked with the online. That's why the online is so appealing to administrators right now.
Yeah, this one guy's got 140,000 students.
Yeah. Well, Mohammed Noor has already taught genetics to tens of thousands of people that he's never met. Now, the taught genetics to might mean something different than what we're used to thinking of as taught genetics to. I have to tell you all it's 1 o'clock, so.
Yeah, I think this is, after answering 30 or 40 questions, I think we're at this point. Thanks so much.